Hello, my name is Larry Dugan. I am a biomedical scientist at Lawrence Livermore National Lab. Today I will be discussing BioID, a rapid and portable system for microbial identification. BioID is a molecular diagnostics platform. Rapid, easy-to-use molecular diagnostics have broad applicability. They have utility in the agricultural space, in food safety, pen-side detection of foreign animal diseases, including foot and mouth disease virus in cattle, and they can be used for the detection of other viruses causing swine and bird flu. The human health arena benefits from diagnostics for the detection of blood-borne pathogens, including organisms that cause sepsis, pathogens involved in urinary tract infections, drug-resistant bacteria, including MRSA, and less common pathogens such as Ebola. Finally, environmental surveillance can also be improved with portable diagnostic platforms. These systems allow for the detection of pathogens from vectors including mosquitoes, fleas, ticks, and rodents in remote settings without the need for large laboratory facilities. So how does BioID work? BioID is an in vitro diagnostic system for the presumptive detection of up to 20 target DNA or RNA sequences from a single sample in 30 to 60 minutes. It utilizes loop-mediated isothermal amplification, or LAMP, to detect pathogen nucleic acid. BioID was developed by Lawrence Livermore National Lab for the U.S. military for rapid detection of biothreat agents in field settings. The system is compact, rugged, and easy to use. It has a small footprint, 2.5 inches tall by 4 inches in diameter, weighing less than 2 pounds. It is man-portable, allowing it to be used in point-of-care, laboratory, field, and low-resource settings. BioID has demonstrated its system functionality from minus 20 degrees Celsius to 45 degrees Celsius, allowing for use in a variety of outdoor settings. Finally, the assay panels are versatile, able to be adjusted to meet a range of detection needs, including human and animal health, foreign animal disease, food safety, plant and pest control, water quality, and vector surveillance. As stated earlier, BioID uses LAMP technology and color change for nucleic acid detection. Advantages of BioID include it's rapid, detecting and providing results in under 1 hour. It utilizes minimal sample prep. It has simple equipment. Since there's no temperature cycling, this reduces the complexity of the hardware. Also, colorimetric detection can be detected by the eye, reducing the needs for sensitive detectors. LAMP itself is an isothermal method for detecting nucleic acid. It was developed by Icahn Chemical Company and was first published on in 2000. This method uses multiple primers for sequence-specific amplification. The addition of loop primers can significantly reduce time to detection. LAMP uses a strand-displacing polymerase that allows for amplification without temperature cycling or initial denaturation of the DNA template, and has optimal activity at 65 degrees. The color change from purple to blue, shown in the figure at the bottom, indicates positive amplification of target sequences. Our working system includes a sample loading device, a syringe, assay cartridge, and a battery-powered heater. The assay cartridges are assembled by a commercial supplier and filled with lyophilized reagent beads at a second vendor. These reagent beads contain all the reaction reagents along with stabilizers for long-term stability. They are sealed in a foil pouch containing desiccant, as shown on the left. The image on the right shows a BioID heater with the lid removed, a reagent bead-filled cartridge having a cap covering the loading port, a 3 ml lure lock syringe that mounts directly onto the loading port, and an FDA card to collect excess sample for future testing. The following video demonstrates the ease of loading a sample onto the cartridge. The white loading port cap has been removed and a 3 ml syringe containing sample has been attached to the loading port, which opens a loading channel. The syringe plunger is depressed, allowing the sample to enter the cartridge and fill the 20 wells. After loading the sample, the cartridge is placed into the heater using a guide pin for proper orientation. The syringe is removed, which reseals the loading channel, preventing backflow, and the white cap is replaced. Next, we place the lid on the lower heating unit and secure it with the rotating lock bar in the center of the lid. Finally, the system is turned on allowing for amplification of the sample. As seen on the right, the display screen is red during initial heating. This turns green during the 60-minute amplification period and yellow following amplification. The screen also displays temperature, time remaining for the reaction, and the battery life. Following amplification, cartridges are inspected visually for color change, indicating positive detection. These images show preloading and post-amplification of a test cartridge. On the left, we see the cartridge placed on an assay identification card that allows us to correctly identify positive and negative reactions. On the right, we see distinct color change in the assay number 8 well, indicating amplification and identifying the presence of this target sequence in our tested sample. Commercial applications of this technology include numerous diagnostic fields, such as human, animal, and plant health, including point-of-care clinical surveillance and outbreak surveillance, food safety, such as microbial contamination and foreign animal disease detection, environmental sampling for soil, air, and water quality, and pathogen vector testing. Target customers might include human health providers, such as medical facilities and out-of-office testing, along with organizations involved in outbreak tracking. Food growers, processors, and inspectors might also use this technology. Air and water quality boards might require testing of samples using these types of diagnostics. And organizations involved in tracking pathogen movement through natural vectors that lead to outbreaks in human and animal health disease might also use this technology. Some of the current practices for microbial identification include clinical and laboratory-based cell culture, PCR-based molecular diagnostics, several of which are FDA approved or cleared, isothermal molecular diagnostics such as Meridian's Illumigene platform, nucleic acid sequencing platforms, and antibody-based diagnostics and lateral flow tests. The BioID system is not a prototype. It is a fully functional molecular diagnostic platform that has been tested over 80 times with DNA and live bacterial cells. We currently have 8 operating units, injection molded cartridges, and over 20 isothermal assays, including assays for biothreat agents, human pathogens including E. coli, Pseudomonas, and MRSA, foreign animal disease pathogens including foot and mouth disease virus, and antimicrobial resistance markers such as MEK and beta-lactamase. Our in-house expertise allows for additional assay development when needed. The BioID system is versatile. Interested users can substitute their own proprietary assays, and temperature profiles can be programmed to meet user needs. We have designs in-house that allow for additional units to be built as needed. Funding on this project is being used to improve reagent bead formulation and develop sensor-based fluorescent detection. This should allow reduction in sample to answer times from 60 minutes down to 10 to 30 minutes. This sensor-based detection will also allow for improved documentation of sample testing results. For more information, please contact the Lawrence Livermore National Laboratories Industrial Partnerships Office at ipo-support@llnl.gov. Thank you.